So today I'm going to talk about something quite different from what we've heard for the rest of this conference is lateral flow immunoassays as a diagnostic tool. So effectively, lateral flows, for anyone who's not familiar, are a really crucial tool in the tracking of infectious diseases, just because they're so low cost, so mass deployable. But ubiquitous to 99% of lateral flows that are out there are antibodies, and they are involved in having antibodies coated onto nanoparticles that give you that colorful signal. So this is typically gold nanoparticles or latex beads. And then we have antibodies that are printed onto our nitrocellulose membrane. So this is our sort of primary antibodies that detect our antigen of interest, and then here is a secondary antibody that gives us that control line. Oh, close to the mic, okay. So antibodies are brilliant in this context because they physisorb directly onto gold, and they can be printed, and they work without any modification to their structure. Just innately, they work really well. However, as Jeremy alluded to yesterday, they're not without their complications. We have issues of batch-to-batch variability with antibody production. Thermal stability is pretty poor, so when we mass distribute these tests, that can be difficult for their long-term stability. Immunogenicity here is also a problem. Because we have secondary antibodies in our blood, and that can cause cross-reactivity issues and low specificity in some lateral flow cases. The price is also an issue. It costs a lot to screen antibodies, but it also costs a lot if you rely on commercial suppliers. Ethical concerns of obviously injecting animals to generate the antibodies in the first place, and you have less flexibility with protein engineering, which is what Jeremy alluded to with the Ankyrons technology. Okay, so the real question was, can antibody alternatives offer improvements to lateral flow immunoassays? So antibody alternatives come into many different shapes and sizes. I just wanted to go through some that have been used in lateral flows before. So nanobodies have been used in lateral flows. They're small alternatives to antibodies that are naturally produced in camelids, so camels, alpacas, llamas. You can also improve them via phage display, which I'll come on to later. And these have been advantageous because they're much smaller than the conventional antibodies, and they have been shown to be a bit more robust as well. And then another alternative is, of course, aptamers. So these are DNA, RNA-based that are selected by many different processes that I'm not going to talk about today, but it's this kind of idea of you just generate a huge library and see what binds to your target. And these have been shown to work in lateral flow, but purely more in an academic basis at the moment. And then what I'm going to talk about today is what we use, which is affinity proteins. So these are small proteins, typically small, that are naturally occurring, and you make mutations to them so they bind to new targets. So Ankyrons are an example of this, which has led to the Ankyrons technology that Jeremy talked about yesterday. What I'm going to talk about today is affibodies, which is what we selected to use. But effectively, these are really useful because they're fully synthetic, and they can be reduced recombinantly with lots of different protein modifications. So a little bit more detail on what affibodies are. This is an affibody dimer structure here. So one single monomer has 3 helices. Effectively, they're based on the structure of protein A. So protein A recognizes IgGs. There are 13 amino acids associated with that strong interaction. If you mutate those via phage display, you generate a large library of possible affibodies, and you take out in the process, you then remove affibodies that bind to your target. They've only really been used in the context of therapeutics, very sort of heavily focused on cancer, some on arthritis, and some imaging applications as well, but purely just more in the oncology space, so not been used in infectious disease or in diagnosis. So we were really interested in taking some of these advantages that I've got listed here, and applying that to lateral flow because we felt that could be really important for low-cost, mass-deployable diagnostics that don't rely on any animal models. But there's obviously a catch with all of this stuff. We're competing with millions of years of evolution here, so there is no guarantee that you will find an affibody for a particular antigen. That's always a disadvantage of this approach. You sometimes get amazing hits, you sometimes get absolutely nothing. So it's a bit of, you need multiple different types of technology to employ here. Okay, so what I'm going to talk about today is a paper that was published earlier this year, where we showcased going from affibody selection to an antigen. We chose the spike trimer here. This was a COVID project. So we go all the way from the selection through to screening, and then we show we can go straight down to an antibody-free lateral flow immunoassay. So that's the story I'm going to go through for the rest of this talk. Okay, so affibody screening. So phage display is a really fascinating process. It's something that I didn't actually do in this work. We did this via a collaborator. But effectively what you do is you generate a massive library of bacteriophages that present the affibody on the surface, on the head. You go through many cycles where you up the stringency every time, so you make it more and more difficult to bind to your antigen. And you effectively only leave the ones that bind to your antigen at the end. You then do what's called a phage ELISA, but effectively what you're looking there is just to make sure that your binders are specific. They're not just binding to things that are in your culture or the beads that you immobilize your antigen on. And then you do an SPR screening of those binders that do work to check their binding kinetics. And effectively, we ended up with 18 binders from this selection process. We then chose 3 that we felt were the most interesting from their SPR curves, which are listed here. So they have these kind of nice, either fast K-ons or no K-offs, which are going to be quite interesting for lateral flow immunoassays, as I'll get to. But effectively, what we did here is we chose our 3 best binders and produced them as dimers. We typically always do this for avidity effects, but actually what we found later, which I'll get to, is that dimers actually work much, much better than monomers for reasons I'll get to. Next slide. Okay, so now in the early stage of this project, we had no idea what was going to work. We had no idea whether affibodies were going to work as captured reagents, so on the paper or on the particles or both. So therefore, what we elected to do was do a mass screening just to see if they work in complementary with antibodies or whether they work just on their own. Now, when you have this, even with just with 3 affibodies and 3 antibodies, you then end up with a huge amount of different combinations. So effectively, what we decided to do here was do an ELISA. But the key difference to a normal ELISA is we use a platinum nanocatalyst instead. It works exactly the same way as HRP. It catalyzes the conversion of TMB into a colorful substrate. And what this means is it's a really, really nice technique because it means we can screen antibodies or affibodies on particles, which is very similar to what they'll be in lateral flow, and then we can screen them immobilized onto plastic. So through this screening, we then get this kind of signal-to-noise map, and we can highlight the ones in bright green are the ones that work very well, and then the ones in black are the ones that don't work at all. So it's interesting that if we go back, that we can see that this affibody here that looks pretty good doesn't work at all when we actually start to use it in practice, which is interesting. So it's not a given that SPR will be an indicator of something working well. But conversely, affibody B worked extremely well, and we took that forward to the next step. So then now we have a smaller cohort of combinations. We then do a screening with lateral flow. So therefore, what we do here is we're printing our capture reagents onto our tests onto paper, and then we're physisorbing our detection reagent onto the particles. And effectively, we then do exactly the same screening, but with lateral flow. And then we take forward our best candidates. Now, something I wanted to allude to here is something that surprised us in this work, which is something that I think is something we really should look into a bit further, is we added a cysteine to the end of our affibodies in this work as well. And this was because cysteines are known to coordinate well to gold nanoparticles or platinum nanoparticles. In this case, it didn't make a difference with the performance when it's on the particles. However, if you compare this one here and this one here, when we add the cysteine, for some reason, printing it on paper made it jump in performance by a huge amount. And we have no idea why, but it just goes to show that this kind of protein engineering approach that you can do with these kind of binders can work really well. Okay, so then we finally took forward our best performance from this screening and did a further analysis. And the sort of take-home message here from all of this data is that if you just combine just affibodies or just antibodies, you get exactly the same performance. And we optimized all of these to try and give it the fairest comparison. And we found that they perform just as well, either on their own, just affibodies, or just antibodies. So this was really nice. You can see there's some slight differences in the curves here, but when you start to analyze the statistics, there's no significance there. So this goes to show that basically affibodies in this context were a direct replacement. And the key thing is is that here we're just doing industrial processes. We're just printing them onto paper. We're just physisorbing them onto particles. We're not doing anything different, no fancy protein engineering. We are just doing a direct replacement for antibodies. This is really a positive. So concluding the talk, we then wanted to show a real case example. So what we did here is we diluted the spike antigen into human pooled saliva. We then used gold nanoparticles instead of platinum because they're much more common, and we wanted to show the sort of versatility of this approach. We wanted to do this antibody-free, so therefore what we did is we have here as our control line is streptavidin, and then we had a biotinylated affibody as our capture instead. So that means that you basically will always form a test line, as you can see here. I mean, in some cases, the very, very high antigen concentration, you don't see a control line, but that's not a really physiologically relevant concentration. And that means now we have a completely antibody-free lateral flow immunoassay. The sort of cherry on top of the cake, as I alluded to at the beginning, is that affibodies are much more stable than antibodies. And we showed this by having our antibodies printed onto lateral flow strips, and then we show that if you store them under the usual conditions, you can see the stability is good compared to day one. But then when you start to store them under sort of challenging, like accelerated aging conditions, high humidity, just on the bench, you can see our affibody is outperforming the antibody in all cases. So this is a really nice proof of concept work that shows that these animal-free derived binders can form a direct replacement to antibodies. You know, what I will say is this is a work with one antigen, and as we know, when we change antigens, things will change. So, you know, there is more work needed to show that this is ubiquitous across different antigen types, different targets, but I think it's a nice way to show that we can move away from antibodies for in vitro diagnostics. So just with that, I would just like to thank you all for your attention. A big thank you to all the authors on this paper, especially Christy, who was a PhD student in the group, who we worked closely together on this work, and Molly, my supervisor. And yeah, thank you so much. Any questions?
So in terms of affibody generation, then, what's your success rate? And have you tested them against any sort of relevant sort of other disease antigens?
Yeah, so we don't do it in our group. We collaborate with a group in Sweden that does this. But in terms of the success rate, so we were working on a cholera project, and we tried to do LPS for the cholera bacteria. That didn't work. And it's hard to know whether that's because of the limitations of the affibody or the way that the phage display works, in terms of the fact you have to immobilize the antigen on either these test tubes that you have these sticky surfaces, or you use sort of magnetic beads or streptavidin beads. And that didn't work. And then we've had other successes, though, with cancer antigens. They work beautifully, but it's hard to make a case for cancer when it comes to lateral flow immunoassays. So the applicability of those is a harder sell. So now it's more about that we really want to move on to infectious disease antigens, but phage display is an expensive process, so it's justifying the resources that go into that. So that's the next step now for us, is to try and work on doing more screening, or moving on to other affinity reagents.